I'm George Mellick, Deputy Director of the Eskitis Institute for Drug Discovery here at Griffith University. I'm also the lead in the Queensland Parkinsons project. So Parkinson's disease is a degenerative disease of the brain, and it affects about 80,000 Australians, and it's estimated to cost our economy around $10 billion annually. There are currently no treatments to prevent, to reverse, or to cure the brain death that we see in Parkinson's disease. The first main problem is that we have no idea as to why those brain cells die. So we need to find that out before we can hope to find a cure for the disease. This is where the Queensland Parkinsons project comes in. The project's a register of over 4,000 people who have agreed to participate in research into Parkinson's disease and related disorders. First, we collect and store important samples from people with Parkinson's, including their blood, tissue samples, and we extract from those things DNA for study of genetics. We also collect very important clinical and risk factor information that's used to integrate into our studies. Here at the Eskitis Institute, we have three important resources that enable us to integrate our approach to studying Parkinson's disease. The first of these is our Queensland Parkinsons project biobank, which includes all of the blood samples and the DNA from our Parkinson's participants. The second resource is the neurobank, which contains over 200 cell lines derived from people with neurological diseases, including over 60 from our Parkinson's patients. The third resource is known as the Eskitis Nature Bank, which contains over 63,000 biological specimens representing the natural diversity of Australia and the Asia-Pacific region. The Nature Bank consists of plant material, marine organisms and microorganisms, all of which have been collected and carefully catalogued and processed in a certain way that enables us to derive or generate the potential drugs from these molecules. One of our strongest assets is our ability to be able to integrate different disciplines of research into one common theme. So we bring together people who are studying the genetics of the disease and the clinical science with the chemists who are able to actually design and synthesize new drugs, the biologists who can study the biological processes, and the bioinformaticians who can put all of the data together into one place and give us a good integrated answer to questions that are very complicated. I think that's one of our biggest advantages. Our study of genetics is actually identifying new targets for potential treatments, and I think this is a major advance, and one of the things that we in the Eskitis Institute are contributing greatly to. We have to be able to accept the challenge of identifying new biomarkers, and this is something we're also working on. But together with new targets and with biomarkers that can tell us about the disease process, I think we're in a very good position to identify new treatments that won't just treat the symptoms of disease, but actually the underlying biological processes. And this is what you need to effect a cure. I can see this happening in the next 10 years.